So Kurt actually gave my talk this title, and I thought it was a good one. So how environmental variability and changes are regulating microbial diversity in the N-bus and in the Gulf of Mexico dead zone. And so I added that part though. So originally I was just going to talk to you about the N-bus, but I added the ... means that I added to the title and in the Gulf of Mexico dead zone. So I'm going to be talking about two different low oxygen environments in the marine system. And I have keywords that I'm going to define as we go through the talk. So they're listed here, but one that I right now want to go over is when I say microbe, what do I mean when I say microbe? So in this talk, a microbe refers to bacteria and archaea. And archaea might be a microbe that some of you are unfamiliar with, and that's okay because we're going to actually talk specifically about archaea today, and we'll learn about that group of microbes. So they look just like bacteria. They're indistinguishable in terms of what the cell structure looks like. They're the same size, very, very small. You can only see them with the microscope, but they're actually a separate domain from the bacteria. Okay, so let's go ahead and get started. All right, so why do microbes matter? Well, microorganisms may be the smallest form of life, but they are actually the bulk of biomass on Earth. They carry out many necessary chemical reactions for higher organisms. In the absence of microbes, higher life forms would never have evolved and could not now be sustained. So what do I mean by that? So what do microbes have to do with higher life forms like humans that one day show up? Well, if you think back to early Earth's history over 3 billion years ago, one type of microbe arrives on the scene, and that is a cyanobacteria, which is a type of bacteria that carries out photosynthesis and produces oxygen. So before that, oxygen was not present on planet Earth in appreciable amounts, right? So oxygen begins to accumulate from this photosynthetic reaction. And oxygen is, as we all know, because we all carry out aerobic respiration, we know that aerobic respiration is the most energetic form of respiration, right? So we all use oxygen to carry out respiration. And so that led to an explosion in the diversity of life on planet Earth early in the Earth's history. So we rely on these microbes today even to produce the oxygen from photosynthesis that we need to sustain life as we know it, right? So these microbes are incredibly important to human health. So you could think about this, take a breath in, right? Everyone's breathing in that oxygen. And so what you're actually breathing in is the remnants of past microbial activity or metabolism, right? So you are, of course, breathing in oxygen that's also evolved by plants, but much of the oxygen that we require is coming from microbes in the ocean, right? So photosynthetic microbes that produce oxygen. We also need to appreciate that humans, plants, and animals are intimately tied to microbial activities for the recycling of key nutrients and for degrading organic matter. So let's think about that for a minute. So nutrients would otherwise be lost from the system, except for if it weren't for the actions of microbes. So they are actually master recyclers, right? And so they take those nutrients that are locked up in the organic form and they remineralize organic matter and they release those nutrients back into the inorganic pool where they are then available to sustain additional primary productivity. So they're really important in terms of recycling nutrients and for degrading organic matter that would otherwise be lost from the system. So it's safe to say then that no other life forms are as important as microorganisms for the support and maintenance of life on Earth. Okay, so let's stop for a minute here and think, what do you know about microbes? All right, so everyone pause for a minute. And what is the first thing that comes to mind when you think about microbes? Probably it's disease, and specifically it's probably this illness, right? So we're in the middle of a coronavirus pandemic, And so many of you probably thought, what do I know about microbes? I know that they're really, really annoying. They're causing this global pandemic. And so this is the virus, coronavirus. And so that might be one of the first things that you thought of. And I can completely understand why that could be the case, right? So this is what, you think about microbes, but we have to stop and think for a minute about how important microbes are to us, right? So if you think about it, there are 100 trillion cells in your body, and only 1 in 10 of those is actually a human cell. So here we have, for every human gene in your body, there are 360 microbial genes. So here is your gene. This is us, the human, and this is them. These are the microbes. So you have millions of microbes to trillions of microbes that live in your gut, and they vastly outnumber you, right? So you are, this is a nice artistic rendering of what the human body looks like. Microbes are on the inside and on the outside of the human body. And so it's fair to say that you are mostly microbes. So here, here I fixed this for you. So this is true that we are mostly microbe, right? So microbes in you, the microbes in your gut are incredibly important because you could not derive the nutrients that you need from the foods that you eat. So I've just had breakfast. Some of you have had lunch and maybe even getting close to dinner, if not already there. And so you ate food, and your body has ways to digest that food, but you need help from microbes to do that, right? And so you wouldn't be able to derive the things that you need from your food if it weren't for the help of microbes. Now, you even have a microbial aura that you carry with you everywhere you go. And so this is an animation that shows how much of the human body is actually microbe, right? So we carry microbes on our skin everywhere we go, and we leave these behind. And actually, some people are starting to use your microbial aura that you leave behind as part of forensics, right? So you can tell about the human, and particularly their gut microbiome. You can tell things about humans, like whether they're a vegetarian or not, or whether they're taking antibiotics, because that really impacts your gut microbiome when you take antibiotics. So this is on the inside of the human, where their gut is colonized by microbes, but these microbes are also on your skin. And an important part of your health is your microbiome, right? And so we all learn that when we take antibiotics, that we can sometimes influence our microbiome in a negative way, right? Okay, so you carry your microbial aura everywhere you go. But let's keep thinking about how important microbes are to you. Okay, so this is may seem like a little bit of a tangent here to talk about mitochondria when we were talking about marine microbes, but it's worth thinking about. So this little cartoon says, I've got the power. And so why is that saying I've got the power, this lifting weights, right? So this is a mitochondria. And if you remember back to what mitochondria does, this is where you produce energy. So eukaryotic organisms produce energy in their mitochondria. All right, well, how does that relate to marine microbes, or microbes in general, and microbes and you? So these mitochondria that you rely on to produce energy that your body needs to run all the necessary reactions is a former bacteria, right? So this formed an endosymbiosis with its eukaryotic host, lost most of the genes that it would need, if not, the great majority of genes that this former bacteria would have needed to live without a host. It can no longer do that, but it has a happy home. It has a host, and what it does for the host is produce energy, right? And so thinking about mitochondria, these are actually replicating completely independently of their host. These mitochondria have a genome that is distinct and completely different than your own genome. And so there are some people that think that maybe we should actually recognize these mitochondria that we can't live without as bacteria, right? And so that is true that they replicate independently of the host and they have their own distinct genomes. Okay, so again, going back to, you think microbes are bad guys? Well, maybe we need to reconsider that. So some of these things I've already talked about, that they keep us healthy. We are mostly microbe. And the microbes, the good microbes that are in our guts and on our skin are really important because they actually prevent illnesses, and they prevent the bad bacteria from taking over, right? They make the air breathable. We've already talked about oxygen. They provide new medicines. So we get our antibiotics and other medicinal, they have other medicines that you can derive from microbes that are really important to human health. They help us digest our food. So we've already mentioned that. They keep our environment clean. So many years ago, we had a large, massive oil spill in the Gulf of Mexico, and I studied the microbes that will actually just happily eat oil as their food source, right? So they help clean up pollution in the environment. They support our crops. So for example, bacteria and archaea are the only organisms that can actually fix nitrogen, so atmospheric nitrogen, right? So we rely on those microbes to fix nitrogen and to support plant growth. All right, so we need to maybe think again about microbes being all bad guys, right? So they're not all pathogens. But this is a talk about marine microbes, right? So bacteria and archaea. So let's contrast the ocean microbiome to terrestrial ecosystems. So in the ocean, marine microorganisms are the most biomass in the ocean. They're the most numerous group of organisms on the planet, right? Because if you think about the planet Earth being mostly ocean, right? So mostly marine. Well, we then can begin to appreciate how abundant marine microbes actually are, right? So that's from the surface water column all the way through the sediments and even into ocean crust. So they're the most abundant group of organisms on planet Earth. You'll find marine microbes thriving in all oceanic environments. So that's from below the seafloor, which is ocean crust. And that includes the deepest part of the ocean, which is the Mariana Trench. So nothing is really thriving there except for microbes, right? And you find microbes from the deepest part of the ocean to the ocean surface. Marine microbes are involved in virtually all geochemical reactions that occur in the ocean. And so this is giving you an overview of many of the roles that microbes play in in biogeochemical cycling. So you have microbes that are interacting with phototrophs. You have microbes that are heterotrophs, that are degrading organic matter. And what we're going to focus on today is actually this group right here. So these are ammonia oxidizing bacteria and archaea. So we're specifically going to talk about ammonia oxidizing archaea. And so this is a group that we'll focus on, and we won't be talking about any of the sediment microbial processes, but in previous RGNO talks, you've learned about sulfur oxidizing bacteria. All right, so this is the tree of life. So this isn't just organisms microbes that you would find in the ocean. This is the entire tree of life. And so we're in here somewhere, and we have this incredible diversity of life on planet Earth. Now, what I wanted to show this because I'm hoping that you'll begin to appreciate how diverse microbial life is. So all of these groups represent the bacteria, right? And then all of these groups here represent the archaea. Now you might be wondering, well, what does that red dot mean? So you see a lot of red dots for these archaeal groups here, and you see a lot of red dots with these bacterial groups. So what does that mean? Well, it means that we can't grow a single representative of this entire group of microbes in the lab. Well, why is that a problem? Well, if you can't grow a microbe in the lab, how do you study its physiology? How do you learn what it's doing metabolically, right? And so that's why we have to turn to molecular techniques where we don't have to grow a microbe in the lab. to determine what its physiology is. But if we step back from that, we also just want to know what microbes are there and what their abundances are in the marine environment. So this is actually a book, and I like the title. It's Marine Microbial Diversity, the Key to Earth's Habitability. That's a hard word to say. And so why I like this is because we've already been talking about how important it is that microbes led to the evolution of higher life forms from oxygen production and sustain those higher life forms from oxygen production and from other important reactions. And so if you could catch a glimpse of the microbes in a drop of seawater, which is shown in this spoon, you would begin to appreciate how abundant microbes are in the water column. So in this drop of seawater, this is probably a little more than a mil, maybe a mil, you would find that there are up to 10 to the 6 microbial cells per mil of surface seawater. And so in this drop of seawater, you have an abundance of microbes, and these are, and you also see a diversity of different microbes or different morphologies. But one thing we have to appreciate is that we can't really learn very much about this water sample when we just look at these microbes using epifluorescent microscopy, right? So we can count them, we can get how, an idea of how abundant the microbes are in that sample, which gives us information like this, or cell abundance. But we can't say, well, what is this one doing? What is this one doing? Are these the same, right? So we need to use molecular techniques to understand, who's here and how abundant are they and what are they doing? All right, so the first thing you want to think about, and particularly in the context of this talk, is how do ecosystem disturbances influence microbial diversity? And if you do influence microbial diversity, or even what microbes are there and how abundant they are, do those changes in diversity even matter, right? So those are the questions that we need to be thinking about when we're going into a system and we're trying to learn about the microbes that are there. Okay, so the ecosystem disturbance that I'm going to talk about today is low oxygen in the water column. So you could also refer to these as dead zones and oxygen minimum zones, or OMZs. So I like this because this really captures it well. Ocean deoxygenation, or the loss of oxygen, is everyone's problem, right? So this is from the tiniest microbes in the ocean to the largest fish in the ocean, right? So this is everyone's problem. This also impacts the terrestrial environment, right? And so you have there's a real consequence to ocean deoxygenation, which we're going to talk about. And so this is a figure, it's a heat map. So when you're closer to cold colors like blue, that means you have lower oxygen concentrations. And when you're closer to warm colors, such as red, that means that you have high oxygen concentrations. And we can see that there are several locations. So we're going to talk about this one today, right? This is the NBUS, Northern Benguela Upwelling System. And I'm also going to talk about the Gulf of Mexico right in here. So here's the United States here, and here's where we study this dead zone off the southeast, in the southeastern part of the United States in the Gulf of Mexico. So we have many locations in the world ocean where we have low oxygen conditions. Okay, so this is a figure that shows the loss of oxygen, or ocean deoxygenation, over time. So this is from 1960. This is again another heat map. And so if you have lower oxygen concentrations, a decrease in oxygen, then you would be closer to red, right? So we can see that there are a few places that have actually seen an increase in oxygen, but for the most part, the ocean has seen a loss of oxygen. So oxygen has declined in most of the world ocean. So here are some terms that I said I would define. So what is a dead zone or a hypoxic zone? These are areas with dissolved oxygen, or DO, concentrations that are below 2 milligrams per liter, or 62.5 micromoles per kilogram. So those would be dead zones or hypoxic zones. Dead zones are low in dissolved oxygen, but they are also enriched in byproducts of microaerophilic. So those are microbes that carry out their reactions at lower oxygen concentrations, and those that carry out their microbial metabolisms with anaerobic conditions, which is where you have no oxygen present. So these are low in dissolved oxygen and enriched in microbial metabolic products like carbon dioxide, methane, and nitrous oxide. So let's think about these three gases here for a minute. These three gases are all important greenhouse gases, right? And so we're going to talk about nitrous oxide today. So remember that these OMZs are enriched in these potent greenhouse gases. And in some places, they're also enriched in hydrogen sulfide, which is toxic. All right, so deoxygenation of the ocean is one of the primary consequences of global climate change. Warming waters are expected to enhance stratification and decrease oxygen concentrations. And we can also just think about this in terms of gas solubility, right? So the warmer the water, the less soluble gases are, and that includes oxygen. OMZs are predicted to increase their expanse and severity over time, right? So these are, this is one of the primary consequences of global climate change. And let's go back for a minute here. They're one of the primary consequences of global climate change, but look what they're enriched in, right? So these potent greenhouse gases. All right, so we're talking about low oxygen environments. And so this is up here at the top of this inverted pyramid is 100% oxygen saturation. But we're talking about environments where you start to go down to 2.0 mgs per liter. And what happens when you start to drop below a certain oxygen concentration or saturation is that fish, if they can leave, they'll leave. You'll find that shrimp and crabs are absent. And this continues this environment continues to deteriorate for larger organisms. So you have stress fauna that emerge from sediments. You then reach a point where you have mortality. And at some point, if you reach low enough oxygen concentrations, you have hydrogen sulfide building up in the water column. Again, this is toxic. And when you reach a point where there's just no oxygen, there's no macrofauna that can actually survive this. And so this is a very visible manifestation of low oxygen in the water column. So example here is St. Helena Bay in South Africa, where you have this massive lobster die-off. In the Southeast United States, we also have low oxygen conditions in a variety of locations, actually. And so this is showing what's called a crab or a fish jubilee. So what's happening here is that these crabs in the water column cannot acquire sufficient oxygen to carry out their metabolism. So they actually flee the water column in large numbers. It's a crab jubilee. And you find that the shoreline is just full, of these crabs that are trying to find oxygen. Now, this is off the west coast of the United States. This is in Oregon, and this is just north of California. And there is an oxygen minimum zone that is expanding and increasing in severity. It's lasting longer, it's getting bigger. And in that environment, you actually have hydrogen sulfide that is building up in the water column, which is toxic. And that led to these crabs dying and then washing up on shore. So the birds actually were pretty happy about this because this represents a nice buffet for them. So it might be hard for you to see, but these birds are actually feasting on these dead crabs that washed up on shore that were killed by the toxic hydrogen sulfide that began to accumulate because of that low oxygen condition. So this is a very visible manifestation of low oxygen. And we're going to talk about two different systems today. So the first is in the Gulf of Mexico. So this is showing you a portion of the United States. So this is Florida right here, and I am currently somewhere about here. And what it's showing here is this darker blue line is the Mississippi River. And this drains a large portion of the United States. And so what's happening here is this Mississippi River is passing through really rich agricultural land. And what we do with agricultural land is we apply a lot of fertilizer, right? So we apply a lot of nitrogen and phosphorus to increase crop yields, right? And so that nitrogen and phosphorus starts to accumulate in the Mississippi River, and eventually this reaches the Gulf of Mexico. And so what happens here is the Mississippi River inputs those nutrients into the water column, and it fuels a phytoplankton bloom. right? And so here's the series of events that happens. You have this fresh river water that's inputting a lot of nutrients here, a very high concentration of nutrients. And then I don't know if any of you are familiar with this part of the world, but it gets really hot here in the summer, right? It's extremely hot. And so you have this heat that is warming up the surface water. You have input all of these nutrients, and then you have an algal bloom. So you have this freshwater lens that stays on top, and you have the deeper, cooler Gulf of Mexico seawater on the bottom. And these stay completely stratified throughout much of the summer. So you have this algal bloom that forms, and then you have these cells start to die, right? These algal cells die, or phytoplankton cells die. And as they're falling to the seafloor, microbes are respiring that biomass, right? That decaying biomass, and they're using up all of the oxygen. And so because this stays stratified, you have what is called a dead zone here, where you have low to no oxygen in this seawater. Now, the Benguela current system, the NBUS that I'm going to talk about, is different, right? And some of you are probably experts in this environment. And so you know that this is actually a very different system, where you have upwelling that provides nutrients, you have a phytoplankton bloom, leading to one of the most, if not the most productive ecosystem in the marine environment in the NBUS. And you have the phytoplankton cells that bloomed, those cells die and they start to follow the seafloor, and you have microbes that are remineralizing or respiring that organic matter, and they're drawing down the oxygen. And so you have this oxygen minimum zone that forms. And so these are actually very different in terms of the mechanism that forms these, you have nutrient input from river in the Gulf of Mexico, and you have upwelling that supplies those nutrients. But both end up at the same place, right? They both end up where you have microbes that respire and use that oxygen, and you have low oxygen minimum zones. So two different systems that end up at the same place with the same problem. Now, we talked about how there is such a strong visible manifestation of changes in oxygen in terms of larger organisms. So I'm just going to skip back there for a sec. So here we go. We have this, very clear signal that there's low oxygen. But what about microbes? Well, you don't really have a visible manifestation of changes in microbial diversity when oxygen is low, right? You can't see these microbes. So is anything even happening to these microbes? Does anything change? Well, one thing that you could observe is that when you have when you have oxygen is replete, organisms that live here are going to use, microbes are going to use that oxygen. But as you start to reduce your oxygen concentration, you're going to turn to different electron acceptors. We're actually going to focus on oxygen consumption today throughout this talk, though. Okay, so then if you could zoom into this particle of, so this is probably a nice, delicious decaying phytoplankton cell or some sort of aggregate of cells. And in this particle, you would actually find that there's an oxygen gradient. So you might have oxygen being replete outside of this particle. And then as you go to the interior, you start to run out of oxygen, and you have a whole diversity of microbes that are going to be found to be abundant in different parts of this particle. But today, what we're actually going to talk about are these cells here. They're not actually associated with the particle. They're ammonia oxidizers. So they breathe ammonia with oxygen. All right, so they're chemolithoautotrophs, and they can fix carbon dioxide, but they're going to do that, and they're going to get their energy through chemical energy, right? So they are going to use ammonia, and they're going to respire it with oxygen, and they're going to generate the energy they need to fix carbon dioxide. So we know that oxygen is one of the primary drivers that actually structures microbial communities. So it determines what that community looks like, which microbes are abundant. It influences diversity, and it influences who's there and how abundant they are. So although there's no visible manifestation of changes because of low oxygen, we know that there are many changes in the water column in terms of the microbial community. All right, so I've already gone over this in terms of the sequence of events here, but here's the Mississippi River. You can actually see the input from space. And so it's carrying all these nutrients, and you have a phytoplankton bloom. Those cells die and they're respired. So you draw down oxygen in the bottom layer, and you have this stratified water column where you have fresh water on top, and you have oxygen deficient bottom water, that is Gulf of Mexico seawater. All right, so some of you might even know Dr. Lauren Gillies-Campbell, and she was in my lab, and she sampled many times in the Gulf of Mexico dead zone. And so she also participated in the RGNO course in 2017, which we're going to talk about. But these are some pictures of sampling in the Gulf of Mexico dead zone. And so one of the things to think about is, how on earth do you determine the composition and diversity of something you cannot see, right? So you have to use molecular techniques. And so what we do is collect samples with a CTD rosette. And so we collect seawater samples in the Niskin. Some of you are very, very familiar with this. So here's Lauren collecting some samples. And then we actually have to filter that seawater we want to increase the microbial biomass so that we can carry out we can use molecular techniques to figure out what microbes are there and how abundant they are. And so we actually extract DNA right off of this filter that's housed in this plastic case. So what do you do if you want to know what microbes are there and look at their abundance? Well, we actually use one, and if you're so unthrilled to be hearing any molecular biology, it's just one slide, so bear with me. So we use a molecular approach, and we amplify 16S rRNA genes from every microbe that is on this filter. And we use that gene because it's a really good taxonomic marker. So every microbe, bacteria and archaea, have 16S rRNA genes. So it's a universal gene. And so we can use it to tell us what microbe is there and we can look at their abundance. So we use this gene, we amplify it from each of the microbes in this sample, and then we sequence each of these 16S rRNA genes. And then we look at patterns of abundance and diversity. So we have to use a molecular approach because we can't just look at a sample and count and tell you what microbes are there and what they might be doing. So the first question is, in the Gulf of Mexico, how does oxygen influence microbial diversity? So these are samples that Lauren collected in the dead zone in 2013. And so this is a map of the samples. And I hope you can clearly see here that there is this large area where you have very low oxygen concentrations. So it earned the nickname the dead zone, right? So larger organisms are not going to be able to carry out their respiration with the oxygen concentrations in this environment. And so this is a heat map. So the cooler colors indicate lower oxygen. And here we have indicated where you would find hypoxic conditions. So how does this influence the microbial diversity? Well, this is using calculating a Shannon diversity index. And you can pretty clearly see here that when you have higher oxygen concentrations in the dead zone, you have the highest microbial diversity. So again, this is just samples from the water column. It's looking at the microbes that are there, and it's calculating diversity. So the highest diversity where you have the highest oxygen concentrations, and then your diversity starts to decrease with decreasing oxygen concentrations. And that continues to be the trend when you've gotten down to the suboxic level with really very, very low oxygen concentrations to no detectable oxygen. So this means that oxygen is absolutely impacting the microbial diversity in that water column. So what microbes dominate this? And so this, I guess this is a little bit of a busy figure, but let's just take a moment and walk through it. So here we have sorted the samples from the lowest oxygen to increasing oxygen all the way to our oxic samples here. All right, so let's look at this. We have hypoxic conditions here. And what I want to draw your attention to is this group of microbes. So these are called Thom archaeota, which I've shown here, And this arrow indicates which one of these colors you would is representing Thaumarchiota. So if we look at this these hypoxic samples, we can see that we actually have pretty high abundances of Thaumarchiota. All right, so this is a type of archaea that's really abundant. And then as you start to increase oxygen concentrations, you can see that for the most part, this Thaumarchiota becomes less abundant, right? So particularly at this really high oxygen concentration. So Thaumarchiota, and it's okay if you don't know what they are because we're going to talk about them. It's a type of archaea that becomes really abundant in the dead zone when you have low oxygen conditions. So we are showing this again. So we're actually plotting oxygen here, and then we're showing abundance of this Thaumarchiota in comparison to oxygen. And so I hope you can see that where you have low oxygen conditions, you have an enrichment of this Thaumarchiota, or this type of archaea. So this was the 2013 dead zone, and Lauren showed the same thing in the 2014 dead zone. This dead zone actually looked very different than this one, and we'll take a look at that in a little bit. Okay, so we see a similar trend. It's not as clear, but we do see that Thaumarchiota are the most abundant when you have the lowest oxygen concentrations. And then when you reach this higher oxygen concentration, they're just not very abundant at all. And in fact, this group of microbes, these archaea called Thaumarchiota, made up over 40% of the entire microbial community in some of those hypoxic samples that she analyzed. So the same pattern was observed in 2014, which is this figure. All right, so let's then move to the NBUS, because as I said, these are these are very different systems in terms of where the nutrients are how the nutrients are being input. So Lauren participated in the 2017 RGNO, and she participated in the course and also the cruises, and she was able to collect 46 samples, which was a wonderful opportunity. And so here's the map of the samples that she obtained, and here are the oxygen. Here's an oxygen profile, so you can see that a lot of her deeper samples were very, very low oxygen was present. All right, so how does low oxygen influence microbial diversity in the NBUS? So we've already talked about how it influences diversity in the Gulf of Mexico dead zone. And here we see a little bit of a different pattern. So we see that actually the highest diversity is found in these dysoxic samples. And in the interest of time, I'm not going to speculate as to why we observe this, but maybe that's something that we could talk about when I'm done with the talk. Okay, so the point of this, though, is that your diversity decreases to the lowest in the suboxic samples. And so that then the beginning and the end of this in terms of diversity is the exact same that you observe in the Gulf of Mexico dead zone. So higher when there's more oxygen, when oxygen's replete, and lowest when oxygen is extremely low to undetectable. All right, so in the NBUS, oxygen concentration is influencing microbial diversity. Okay, so I'm not sure if you're familiar with this type of plot. So this is an ordination. So if we step back and we think about the data that I'm working on, we have a species table, right? And so in this case, the species are microbes. And so we have thousands of different species of microbes in these samples. And we have our samples, right? We have our samples, we have our species, and we have the species abundances. So how do we actually plot that in one reduced space plot? And so what we do is we carry out ordination, so non-metric multidimensional scaling. So this is a type of ordination. And let me explain what you're seeing here. So each one of these points is a sample. And so if you have points that are close together, it means that their microbial communities are really similar. If you have samples that are really far apart, so let's look at this suboxic sample and compare it to, example, this oxic sample. Well, these are actually very different microbial communities, right? And so they plot really far apart on this. And what is actually driving the changes as you go along, let's say axis 2 here, right? So we go along axis 2, we have something that is leading to changes in the microbial community as we go down that axis. And so one of the things that's driving changes is dissolved oxygen concentration. So you can see the suboxic samples are more similar to each other. The disoxic samples are more similar to each other, but have, of course, overlap with both suboxic and some overlap with oxic. And then you can see that the oxic samples are more similar to each other. Now, I took this plot and I show the exact same plot. I didn't change anything here, except each one of these samples is showing you by size how many Thom archaeota are in that sample. So we have bigger bubbles, meaning that we have more Thom archaeota in these samples. And so you can see that there's more Thom archaeota where you have lower oxygen concentration. So these are the suboxic samples. And the same is true for these disoxic samples. You can see bigger bubbles, meaning you have more of it. And then when you get to oxic samples, you can see that the bubbles are really small. You don't have as much of this Thom archaeota. So you could look at this another way, and you could see that Thom archaeota abundances are increasing when oxygen decreases, right? So what microbes dominate low oxygen samples? Well, remember we learned that in the Gulf of Mexico, what dominates are this type of archaea, Thom archaeota. We see the same thing in the NBUS. All right, so I keep saying you're going to learn about archaea, and I haven't delivered on that promise I made at the beginning of this, right? Well, here we go. So you might not be at all familiar with archaea. Again, they look just like bacteria. They're single-celled, they have no true nucleus, and they're extremely small. They're the same size as bacteria, so you can't see them with your eyes, right? So originally, archaea were thought to be aberrant or weird members of the bacteria. They were thought to inhabit only extreme environments, so were called extremophiles. So where you might find these living would be in boiling mud pots. Okay, so this is a boiling mud pot from Yellowstone National Park, which is here in the United States. This is an amazing place to go and see geothermal features such as this. So this boiling mud pot is really acidic and it's really hot. It's literally boiling mud. So what do you think is really abundant in this environment? What a hostile environment, right? Well, what you would find being very abundant here would be archaea, right? So you would find that there are numerous archaea and they're carrying out some form of sulfur metabolism, right? So this is a really acidic, really hot environment. This would be one place you might see archaea. Another place would be this super hot hydrothermal vent environment. In on the seafloor where new ocean crust is forming, you would find archaea here. So really high temperatures, really reduced conditions in that vent fluid. Now this here is actually acid mine drainage. And so this is pH, I'm gonna pause this. This is pH 0, right? So what do you think is thriving in this environment? Not much, right? But what you do find thriving here are archaea. So this was originally thought to represent the extent of the environment where you would find archaea, these really extreme environments. And you also can find archaea. This is the Great Salt Lake in the United States. This has an incredibly high salinity. So you would find archaea thriving here. This is a seawater evaporating pond, and I understand that the RGNO students are actually going to be able to see this. So what gives this feature this color is actually archaea, right? So, so you might actually be able to see something like that during the course. This is a soda lake, so the pH is 10, and again, that color is coming from the archaea that lives here. And I would be remiss if I didn't mention that you can actually find, this is in a saltern, you can actually find square archaea. So who would have thought that in nature, cells are actually square for their morphology is actually square. I would not have thought that unless I knew about this type of archaea. All right, so what we've learned about archaea is that they thrive in extreme environments, and that's what we thought was the extent of their habitat. But 22 years ago, there was a study that was done at Station Aloha. So that's off of the, that's in the water column off of Hawaii in the United States, right? So the middle of the Pacific Ocean. So here they went from the surface all the way down to almost 5,000 meters and counted the cells there. And so the first thing they did is count bacterial cells. And so you can see bacterial cells really dominate that surface at Station Aloha. So it's this long-term, it's a station that has been visited every year, numerous times a year for a long time now. And so it's generated a lot of data. So 22 years ago, this observation was made. Bacteria are abundant at the surface, but they decrease with depth. But here are those Thaumarchaeota. So they're not abundant at the surface, but they increase with depth. And by 1,000 meters, they're the same in terms of abundance as bacterial cells. All right, so that means that these Thaumarchaeota represent one of the single most abundant cell types in the ocean, and that the global oceans harbor 1.3 times 10 to the 28 archaeal cells. Many of them are Thaumarchaeota. Remember, everybody, this was 22 years ago, and we were still thinking that these are the only environments where you would find archaea. We didn't think they would be thriving in the water column. Now, more recently, it's been recognized that Thaumarchaeota are abundant in coastal environments. And in Lauren's work, she actually found one specific type of Thaumarchaeota in the dead zone that appears to be adapted to low oxygen. And so that's what she's showing here. This is one type of microbe. So here's low oxygen, and this is microbe is really abundant. 2014, the dead zone was very different. It was very shallow and more coastal. But we see again that this one type of Thaumarchaeota is really abundant, and it appears to be adapted to these low oxygen conditions. All right, so we know that Thaumarchaeota are active with low dissolved oxygen concentrations. So there's an accumulation of molecular and isotopic analyses that have revealed that ammonia oxidizing archaea. So let's stop right there just for a second. So these, I mentioned them, at one of the first slides. These ammonia oxidizing archaea are, it's abbreviated as AOA. So they are using ammonia and they're using oxygen, and they're deriving their energy from that reaction to fix carbon dioxide. So they're ammonia oxidizing archaea. And they're really important contributors to the nitrogen cycle because they're using that ammonia, right? So in 2012, they were identified, these AOA, as being one of the dominant energy sources in the Eastern Tropical South Pacific OMZ. And in the NBUS, there have been a couple studies that reported that there's active ammonia oxidation. And if I remember correctly, these were attributed to Thaumarchaeota. Okay, so they're also abundant when there's low oxygen concentrations in the water column. So there's numerous studies that have shown an increase in the abundance of AOA when oxygen is low. That includes our work in the Gulf of Mexico, where some of our samples were up to 40% of this Thaumarchaeota. In the NBUS, Lau et al. Reported that ammonia oxidizing archaea abundances were up to 33% of the microbial community in their low oxygen samples. These were collected in January 2014. Lauren comes along, collects samples in 2017, and this is April to May when the cruises went out. And she also found an enrichment of AOA in the NBUS, and up to 20% of the microbial community was actually this type of Thaumarchaeota that oxidizes ammonia. The highest abundances were in dysoxic and suboxic samples, which is shown here. So here's suboxic to dysoxic to oxic. Okay, so why do we care? Why does it matter if this Thaumarchaeota is really abundant in these oxygen minimum zones? Remember, one of the first things we talked about was that in these oxygen minimum zones, you have an enrichment of nitrous oxide, a potent greenhouse gas. So where does that nitrous oxide come from? Well, there's actually been several studies that have shown that ammonia oxidizing archaea are dominating nitrous oxide production in some regions. And Centaur et al. Reported that ammonia oxidizing archaea, AOA, may actually be largely responsible for the oceanic nitrous oxide source, and that includes in OMZs. So we have a potent greenhouse gas that is being produced as the result of these ammonia oxidizing archaea or these Thom archaeota that are abundant in both the dead zone and in the N-bus. All right, so I know I need to stop, right? But this is the last slide. So what we think about is, we ask the question, does DO concentration impact diversity and community structure in these two different OMZs? And the answer is yes. In fact, oxygen is one of the primary drivers that leads to the structure of that microbial community in both of these systems. And why this is significant if you have high abundances of this Thom archaeota when you have low oxygen is, one, they're aerobic respirers, right? So they're drawing down the oxygen even more. They can carry out their metabolism with low oxygen concentrations. And importantly, they're the likely source of this potent greenhouse gas in these OMZs. Okay, so Kurt said I could just let you know about an opportunity I have in my lab. So I'm going to do that, and then I'm going to come back to this slide. So I just wanted to make the students aware that I have funding for an African graduate student to come to my lab and work with me to learn about how to analyze microbial data. And so this could be your own data or it could be my data. And so it's a paid opportunity where I have 6 months of funding. And here's my contact information. So if you want to learn more about this, obviously this would be something that your advisor would be supporting and happy for you to do. But let me know if you're interested and we can talk more about that. So let me go back to, let me go back to this slide and see if there are any questions.
Is that it, Olivia?
Yes, that's it.
Thank you so much. I thought you would still continue from there. Thank you so much for the presentation, and I'm sure we all have learned a lot. Personally, I lived to respect for microbes until today. So thank you so much for this enlightenment, and I'm sure there will be a lot of questions. Questions? Comments?
Kurt, you always have questions. Oh, it looks like we do have a question. So if I know you have questions, but
it's true, Olivia, but I don't want to be the first one. You know,
we have a hand from Brishan.
Yeah, Nicola has a smartphone, so.
Hi, Olivia. Thank you for the amazing talk.
Yeah.
My question is on the molecular side of things. I just want to know what type of sequencing and stuff was done for the microbial DNA.
Yeah, so that's a great question, and I probably should put that in, but I know I tend to lose audience members when I start really talking about that kind of stuff. So so this was ITAG sequencing. So we used the MySeq, and these were these reads were about 250 base pairs. So we were using the 250 base pair chemistry, and we prepared these amplicons ourselves in the lab, and then used the MySeq to sequence them.
Okay, thank you.
Yep. Kurt.
Yes, thank you, Olivia. Always interested of what you find, and especially if you find things that can be compared between very different environments. Originally, you think they are different, but in the end, they still turn out to be very, very similar. Okay. Now, the studies that you do in Mexico, or Gulf of Mexico, can be applied to the bush and vice versa. Okay. One of your last comments was that you found now Thom archaeota in areas with low oxygen, right? So do you expect them also slowly to become able to oxidize ammonia under anoxic conditions?
That's a good question. And I actually would say the answer to that was probably no. And the reason why I think that is the case is because there was a brand new paper that came out. And this is dangerous because I haven't even read the paper yet, but there's a new paper that came out that showed there's no understanding of the mechanism, but what's happening during their metabolism is that these Thom archaeota can actually evolve oxygen intracellularly. And so they can actually produce oxygen. And so I don't think that there's going to be a complete evolution to carrying out this reaction anaerobically. And I think that because it might not be necessary if these microbes can actually evolve oxygen through their metabolism. But again, there's no understanding of the mechanism by which that oxygen is being evolved.
Are you saying this is a heterotroph making oxygen without light?
These are chemolithoautotrophs, so they can fix carbon dioxide. So actually what I'm saying is, again, we don't know the mechanism, but there was a paper that just came out that said that by some means, they're actually producing oxygen without carrying out photosynthesis.
How does that work? I mean,
we don't know. Yeah, so everyone was just scratching their head with that paper because it's revolutionary, right? And it could really explain why we keep finding thaumarchia to be so abundant in these low oxygen environments. Because, well, one, they actually have enzymes that have a high affinity for oxygen, so they can do that with low oxygen. But if they're actually producing oxygen intracellularly, that just really does, we need to rethink that. I don't understand the mechanism.
Thank you, Olivia. Other questions?
We have a question in the comments in the chat from Cecilia.
Let me see. I have to figure out how to get my. How do you get that back? Would someone actually read the question to me, because I can't get the chat back.
I can do so. I'll do so. Saying are the studies that have quantified the amount of nitrous oxide from upwelling regions or other OMZ regions?
Yes, if the question is, are there studies that quantify N2O production? And the answer is yes, for sure. One of the reasons, so I actually have a new project that was funded by the National Science Foundation here in the US. And we're going to participate in this course, but also sail on a US ship at some point next year. And the project goal is to understand, first, to quantify the nitrous oxide that's being produced in the N bus, and then to try to figure out the microbes that are producing that N2O. So that's actually the whole goal of the project. And there's a lot of modeling that's done of N2O production in OMZs, but the models don't have good measurements in the N bus, right? So we have to use N2O measurements that have been made in other OMZs and apply that to the models that are run to look at N2O production and consumption in the N bus. And so that's what we're trying to do is actually understand, quantify how much N2O is produced and determine what microbes are leading to that N2O production. So that's a good question. And the answer is yes, but it's really not a lot of data. There is some, but not a lot of measurements that have been made for N2O in the N bus.
Thank you very much, Olivia. And then we have a hand by Gaddafi. Please go ahead and. Okay.
Hey, good afternoon.
Good afternoon.
Yes. I have a simpler question here. I see you have done a lot of good work, and it's interesting. And I just wanted to find out, because I have a couple of NGS files or data results that I got from my previous project that I was working on. Mm-hmm. And I'm having difficulties finding an online page to analyze this data, to actually get the organisms or species lists. Yeah. Is there any site maybe that you know that is available for free?
Yeah, so that's mostly you could use something like, it's a US website. You could use something like KBase. What type of data is it actually? Is it the whole, like, is it metagenomics or genomic? I'm not sure what type of data do you have?
Oh, it's NGS metagenomic.
Oh, okay. And is it of the microbial community?
Yes.
Well, I mean, again, I'm open to collaborations to help with that sort of thing, because I do all of my metagenome assembly and analysis on my server, but I recognize that not everyone has access to that or the experience with doing metagenome assembly. So, we could talk separately about that. But if you want to just find something online, you could look into KBase. because you can assemble with K-base. I haven't had great luck with getting really good assemblies from that online tool. But you can check out K-base and you can look into Galaxy as well.
Okay.
But yeah, but we could also just talk separately.
All right.
Just a word to Gaddafi. Gaddafi, please make use of Olivia. She will join RGNO this spring in April. So make sure that you can meet with her at that time.
Yes, we'll surely do, Prof.
Okay, thank you. The next question is, sorry, the next hand I'm seeing is from Foka. I'm saying the name right?
Yeah, you are right. I hope you can hear me.
I'm just capturing, sorry, I'm just capturing the name of the person who just asked me that question so I can follow up with them. Okay, go ahead. What's the next question?
Yeah, it's maybe more a comment. So first one, thank you for your really nice talk. It was very impressive to me, especially since I'm back only 2 weeks from a 7 weeks cruise in the Enbus. And there we made also a lot of microbiological studies. I, for myself, I'm a physicist, and therefore I cannot tell you too much about it. But anyway, we really also looked into N2O production there and made a lot of measurements, including pump CDDs, underway measurements, and so on. So therefore, it seems to me it might be a good idea that we can work together on it.
Absolutely.
Because it's every time good to have more data to see these problems. And what we also found is that we have large amounts of ammonia in the water column, even in regions where we found some oxygen. So there is more or less enough food for these, let's say, how is Sommer care also. I'm not so familiar with these terms. So, and yeah, that's only what that I want to mention. And it's really sad that I cannot participate in the RGNO this year. But I think you will enjoy it, really.
Yeah, no, I appreciate that. I absolutely am open to that. I agree. More data is always better. And working together to compile is great. So I'm showing, I think you can see my email address here. So if you want to email me, that would be great.
Yeah, I'll do so.
Okay, thank you.
Okay, do we have any other? Okay, I think Brishan is back again. Brishan, please go ahead.
Hi. So I just wanted to find out. So I'm not. I don't look at archaea or bacteria per se in my studies, but I wanted to know if you know anything about people looking at microbial communities in non-oxygen minimum zones and what their community compositions are. So like in the Gallus current, for example, instead of the Benguela current.
Yeah, well, I mean, I've done a fair amount of work just looking at the water column in the Gulf of Mexico where we don't have low oxygen. So if you go off the coast of Louisiana down there, down here, and you find very typical microbial communities where the thom archaeota are not very abundant unless you go deeper in the water column. So what you're going to find is really abundant is something like SAR11, which is the most abundant heterotroph on planet Earth, right? And so that's a really common microbe that can be abundant from the surface even all the way to the seafloor. And so that's just a heterotrophic metabolism at this, when you go from the surface down to probably 100 meters. Now, this might not be true for coastal environments, but in just pelagic environments, you're going to find really high abundances of cyanobacteria like Prochlorococcus and Synechococcus would really dominate that surface water column. But yeah, so there's many studies that have looked at the water, the water column outside of OMZs, and you do find that there's very common, a lot of overlap in the microbes that are present, but also abundant. Yeah.
Okay, thank you.
So as an aside, also, I would just say that that there is some thought that SAR11 is closely related to mitochondria. It's really controversial, but I still like to think sometimes that there is a really abundant heterotroph, the most abundant heterotroph in the world that is abundant in the water column that actually lives inside each of us, right? So that's super controversial. Maybe they're not that closely related to a mitochondria, but there is some evidence that they are. And so that SAR11 is maybe a close relative of the mitochondria that we all rely on for energy generation.
Okay, thank you, Olivia. There's a question in the comment in the chat, and that is from Sam Mafila. And it goes as how much data has gone into the distribution maps of dissolved oxygen to depict the OMZs? Are the hotspots moving with time? And then maybe before you answer, if I can just add mine also, my much shorter one. What might be the cause of the expansion in the dead zones that you explained earlier? Why are they expanding?
Right, yeah. So, I mean, the primary reason is climate change, right? Because you're increasing the stratification in the water column. And so you're also reducing gas solubility, right? And so if you're reducing gas solubility, that means that your oxygen is not going to dissolve into seawater as readily, right? So you have, oxygen is obviously, makes up 20+% of our, excuse me, of our atmosphere. And so at the surface, you can have that equilibration. You also have photosynthesis that's going to lead to oxygen being produced. But when you have warmer conditions, your gas solubility decreases, right? And so you have less oxygen, and you actually have oxygen that's, decreasing in the water column in the global ocean. So you have increased stratification where you have those separate layers and you don't get oxygen replenishment. And then you have decreased gas solubility, but then also in the Gulf of Mexico, you have increased input of nutrients, right? So you're fueling that phytoplankton bloom, and then that is fueling that drawdown of oxygen, right? So you have many different reasons, but climate change is the primary reason.
Okay, thanks. Okay, then I think in that case, if there's no further questions, I can see Brishan send his up again, but I think that's good for the discussion. Thank you. Go ahead.
Yeah,
please.
sorry. Since there's no further questions, I just wanted to ask Prof about her opportunity. And is it how soon would one have to apply? And is it a one-time opportunity, or will there be one next year or the following year?
Yeah, it's pretty open. So I received funding, and I don't have a specific start date. It's really up to the student, right, and their advisor, what works. But the idea behind that is to bring that expertise back to the lab, and so that needs to be something that your advisor would be interested in having in the lab, right? So it's to try to increase the expertise in analyzing microbial data. And so that's the idea there. So it's really open, and it could be broken apart in between, it could be 3 months and then another year, 3 months, but it's a 3-year grant. So the money does need to be spent within the next 3 years. Yeah.
Okay, thank you very much, Olivia, and thank you for the questions and indeed for your participation in the discussions. Now, I don't think there's any other I don't see any other hand, and so maybe at this stage I'll give it over to Kate. But before that, let me just thank you for your participation, and indeed for the very good presentation that really, I think it is enlightened me. I'm sure it has done the same for the rest of us. Thank you very much, and Kate, please, it's over to you. Thank you very much.